So we've got a bunch of people still coming in, so we'll let everybody come in.
Sounds
and
good.
start maybe in 1 or 2 minutes. Okay, let's get started. So good morning, everybody, or afternoon, wherever you are. Thank you, everybody, for joining us again for another NextGen Epilepsy seminar series. This week, or this month, we have a really cool talk by Dr. Trina Basu. And Trina is a postdoc in Jamie McGuire's lab at Tufts University, and she's going to talk to us today about sex hormones and or is it sex hormones or just the HPA axis?
Just the HPA axis.
I'm going to learn. I think I'm going to learn today.
Well, for today, it's the HPA axis.
Yeah. And seizures and circuits and behavior and all kinds of good stuff. Trina got her PhD in Aptar Rupert's lab at University of Wisconsin, where she worked on another mouse model of epilepsy, the TSC. It was a TSC knockout mouse. Is that?
It was the TSC2 mice. Yeah.
TSC2 knockout mice. And so, all in all, a very robust epilepsy training. So we're excited to have you here. And so take it away.
All right, thank you. I wanted to first thank Momoko and Bobby for putting on such an amazing seminar series so far. The science has been incredible, and I am honored to be here today to present my work in Jamie's lab. So as my title suggests, I'm going to be talking about how seizure-induced activation of the hypothalamic pituitary adrenal, or HPA axis, worsens epilepsy outcomes and associated comorbidities. So while seizures are the primary manifestation of epilepsy, this disease presents with many comorbidities, including the ones that are shown here. So many people with epilepsy suffer from memory and learning deficits. They have sleep disorders. But most importantly, though, these comorbidities negatively impact a person with epilepsy's quality of life. And in extreme cases, if it's left untreated, such as the case for sleep disorders, if you have sleep deprivation or if you have high levels of stress, it can actually worsen disease progression. So today, my talk will be focusing on mood disorders in epilepsy, and specifically in depression and anxiety. Approximately 25% of people with epilepsy suffer from anxiety, and up to 55% suffer from depression. And I want to take a second here to really emphasize that these really high incidence rates of anxiety and depression in the epilepsy population is not solely because they're living with a chronic illness. In fact, now numerous publications have shown that mood disorders in epilepsy occur at incidence rates that are much higher than the general population. And more importantly, it's much higher than any other chronic illness patient population. So this already kind of starts to hint at some underlying pathology that predisposes people with epilepsy to suffer more from mood disorders. And interestingly, patients that suffer from major depression disorders have exhibited a 3 to 7-fold higher risk of having an unprovoked seizure and or even potentially developing epilepsy. So this fact alone starts to hint at a potential bidirectional relationship between epilepsy and depression. And our lab is particularly interested in understanding this bidirectional relationship and investigating the underlying pathology that predisposes people with epilepsy to suffer from mood disorders. So in order to explore this potential bidirectional relationship, it's best to learn from depression research. This field has been heavily studied and a lot is already known about depression. So we can start to ask questions based on depression research to help us bridge this connection between epilepsy and depression. One of the hallmark features of depression is the overactivation of the hypothalamic pituitary adrenal or HPA axis. So the body's physiological response to stress is mediated by the HPA axis. So in response to a stressor, whether it's a physiological stressor or a psychological stressor, corticotropin releasing hormone or CRH neurons in the periventricular nucleus of the hypothalamus releases CRH. CRH can then go on to activate the pituitary gland to release adrenocorticotropin hormone or ACTH. And finally, ACTH stimulates the adrenal gland, which leads to the release of cortisol. Cortisol levels in the system and release is regulated through this feedback inhibition. And in depression, this feedback inhibition is impaired. And this leads to a pathologically high levels of cortisol or circulating cortisol or hypercortisolism. And these pathologically high levels of cortisol can also be detrimental and can damage the structural and functional integrity of brain regions like the hippocampus, which is of particular interest for people who study epilepsy, because in epilepsy, the hippocampus is also significantly damaged. In epilepsy, between 30 to 60% of people with epilepsy self-report stress as a trigger for seizures, and many also report that it worsens their epilepsy. And similar to what we observe in depression research, cortisol levels are just basally elevated in people with epilepsy. And this is further increased following a seizure. And there have been clinical studies that have shown that people with epilepsy exhibit a loss of the HPA axis feedback inhibition, similar to what has been seen in depression research. So the clinical studies alone suggest that there might be a common pathophysiological mechanism that underlies both depression and epilepsy, and this may contribute to their comorbid appearance. So in rodent studies of epilepsy, we see that if you manipulate parts of the HPA axis, this can lead to changes in epilepsy severity. We and others have shown that exogenous application of the hormones corticosterone or CRH can either accelerate epileptogenesis and or increase epileptiform activity and seizure frequency. So this figure here is from a 2013 paper from our lab. And here, mice were implanted with a corticosterone pellet and then injected with canic acid. And then acute seizure susceptibility was measured 2 hours post canic acid injection. Mice that had the exogenous corticosterone in their system exhibited decreased latency to seizure onset. And they also exhibited increased epileptiform activity, suggesting that exogenous core application actually worsens the HPA axis, or seizure susceptibility, sorry. Furthermore, studies by groups like Tali Barham's lab have shown that chronic stress, including early life stress, can accelerate epileptogenesis. And finally, there have been studies that have shown that inhibiting glucocorticoid synthesis or blocking glucocorticoid receptor activity is protective in epilepsy models. In fact, there's been a very recent paper from the Danzer lab that was released a couple days ago, and it shows that an antagonist specific to glucocorticoid receptors blocks hippocampal pathology that's associated with status epilepticus. So overall, these studies further establish that HPA axis dysfunction in epilepsy brings us kind of closer to understanding what the shared pathophysiology is with depression. Our lab has recently shown that seizures alone can activate the HPA axis, and that changes to the HPA axis function can alter epilepsy outcome independent of stress. So 2 hours after status epilepticus that was induced either with canic acid or pilocarpine, there is this significant increase in circulating levels of the hormones ACTH and corticosterone, and that's quantified here compared to vehicle-treated mice. And this is also followed up with an increase in firing rate of CRH neurons in the PVN. And so to understand what is causing this increase in the firing rate of CRH neurons after canic acid or pilocarpine treatment, Kate did a number of studies and found that actually the increased firing rate of CRH neurons after status epilepticus was found to be due to a loss of the protein KCC2. So this is a Western blot of protein isolated from the PVN. And you can see that in the canic acid treated mice compared to vehicle, there is a reduction in the total KCC2 protein levels, as well as a reduction in the phosphorylated KCC2 levels. So what is KCC2? KCC2 is a potassium chloride cotransporter, and it's developmentally regulated. In the immature neuron, KCC2 expression levels are low, and NKCC1 protein levels are high, and this leads to high intracellular chloride levels, which causes the neuron to depolarize it in response to GABA binding. In the mature neuron, this is reversed. So KCC2 protein levels are increased and NKCC1 protein levels are decreased. And this leads to low intracellular levels of chloride. And this results in the neuron hyperpolarizing in response to GABA binding. So in epilepsy models and in other neurological disorders, when KCC2 is lost, the neurons revert back to an immature state. So KCC2 levels are low when it shouldn't be, and this causes GABA binding to be depolarizing. So overall, our data suggests that loss of KCC2 in CRH neurons drives HPA axis activation after seizures. So with this in mind, we collaborated with Steve Moss's lab here at Tufts University to generate mice that lack KCC2 in CRH neurons. So from here on out, whenever I say KCC2 CRH, if you see this indicates the mice that lack KCC2 in CRH neurons. So after initial characterization of these mice, we see that the KCC2 CRH mice have reduced KCC2 protein compared to our wild-type Cre-negative mice. And Jamie was able to characterize their HPA axis activation. And so under basal levels, our KCC2 CRH mice, the mice that don't have KCC2 and CRH neurons, don't exhibit any difference in circulating cork levels compared to wild-type Cre-negative mice. But when you challenge the mouse with canic acid, in the wild-type mice, the Cre-negative wild-type mice, we see this predicted increase in circulating cork levels. It's something that we've observed. But the KCC2 CRH mice have this hyper-exaggerated response. So they show more than double the amounts of circulating cork levels. And this suggests that our KCC2 CRH mice have a hyperactivation of the HPA axis following status epilepticus. So using these mice, we wanted to know whether hyperactivation of the HPA axis contributes to anxiety and depression-like disorders, and if it contributes to seizure susceptibility in a model of chronic epilepsy. So for the following experiments, I used the ventral intrahippocampal canic acid model of temporal lobe epilepsy. So the field, for the most part, uses and looks at dorsal intrahippocampal canic acid injections. And we chose to use the ventral intrahippocampal canic acid model because the ventral hippocampus is more involved in the regulation of affective states. And Zach Ziegler from the Crook Magnuson lab has shown that when you inject canic acid into the ventral hippocampus, this elicits more pronounced anxiety-like and depression-like behavior. So this graph here is showing the amount of time that a mouse spends in the center of an open field paradigm. So in the open field paradigm, mice are, the amount of time that a mouse spends in the center is measured. And if a mouse spends more time in the center and is more exploratory, that's indicative of the mouse being less anxious. And compared to saline injected mice, both the dorsal intrahippocampal canic acid and the ventral intrahippocampal canic acid mice spend less time in the center, suggesting that they're more anxious. But you'll notice that mice that have canic acid injected in the ventral hippocampus have a more exaggerated anxiety-like phenotype. The sucrose preference test is a test of anhedonia. And so mice are given the option of drinking either regular water or sugar water. And like humans, they would much rather prefer drinking flavored sugar water. So if a mouse doesn't drink the sugar water, it's a sign of anhedonia and is thought to be depression-like behavior. So once again, in both cases for dorsal and ventral intrahippocampal canic acid treated mice, they consume less sugar water, suggesting that they're more anhedonic compared to the saline injected mice. But you'll see that the mice that are injected with canic acid in the ventral hippocampus have a more exaggerated, more pronounced anhedonic phenotype. So we decided to use this model because of its more pronounced affective behavior phenotype in our KCC2 mice. So we first tested the ventral intrahippocampal canic acid model in C57 mice, and very similar to what the Crick-Magnuson lab has shown, chronically epileptic wild-type mice exhibit significant anxiety-like behavior. So just to go over the injection and behavior paradigm, mice are injected with canic acid in the ventral hippocampus and then tested on behavior for 60 days after status epilepticus induction. So in the open field test, you'll see similar to what the Crick-Magnuson lab has shown, I was able to see that canic acid treated mice exhibit significantly less time spent in the center, suggesting that they're more anxious. The light dark box is also a paradigm of anxiety. So a mouse is put into a chamber that has a dark box and a light box, and the amount of time that a mouse spends in the light box is indicative of their anxiety level. So if they spend more time out in the light box, they're less anxious. And our canic acid C57 wild type mice spend more time in the dark box, indicating that they're more anxious. And finally, the elevated plus maze is also a test of anxiety. Mice will prefer to stay in high-walled dark arms as compared to the open-walled arms. And so the canic acid treated mice tend to stay more in the closed arms, indicating that they're more anxious. These mice also exhibit a more pronounced depression-like behavior. So we tested them on the sucrose preference test, and similar to what was observed in the Crick-Magnuson group, canic acid treated mice drink less sugar water, suggesting that they're more anhedonic. So now we wanted to test how this model looked in our KCC2 CRH mice and how it impacted their affective states. So the data is a little bit less robust, but there are some patterns in their anxiety-like behavior that I wanted to kind of go over. So in this graph, the green bars represent the mice that are injected with just saline, and the blue bars are mice that are injected with canic acid. The solid bars are mice that are just Cre negative, the Cre negative wild type mice, and the pattern bars are the Cre positive mice so these are the mice that lack KCC2 in their CRH neurons. So in the wild type mice, we see this predicted decrease in the open field paradigm. We see this predicted decrease in the amount of time spent in the center for those that were treated with canic acid compared to saline. So the canic acid chronically epileptic mice are more anxious compared to our saline injected wild type mice. We also see a decrease in the amount of time that our chronically epileptic KCC2 CRH mice spend in the open field, or the center in the open field. This doesn't differ that much from our wild type chronically epileptic mice. And this is suggesting that there may just be a ceiling effect in terms of how anxiety states may manifest with regards to HPA axis activation levels. But if we were to look at the data in a slightly different way, we observed that there was a trend for the KCC2-CRH mice to underperform or be more vulnerable to anxiety-like behavior in this paradigm. So we see that more of the KCC2-CRH mice tend to spend less time in the center compared to the chronically epileptic Cre-negative wild-type mice. So when you graph it up in a pie chart form, you can see this trend a little bit more clearly. So our KCC2 CRH mice, chronically epileptic CRH mice, are more vulnerable. So they spend less time in the center, suggesting that they're more anxious than our Cre-negative wild-type canic acid-treated mice. So the light dark box is also another test for anxiety. And while there does seem to be a general reduction in the time spent in the light box for our KCC2-CRH mice, it's not significant. And you can see there's quite a bit of spread in our data. But once again, if we look at it slightly differently and see how many of our mice are more vulnerable or more susceptible to anxiety-like phenotypes, we'll see that the KCC2-CRH mice tend to spend, a greater number of them tend to spend less time in the light box compared to our Cre-negative canic acid-treated mice. And that's graphed up here. So you can see generally there seems to be a trend for the KCC2-CRH mice to be more vulnerable and underperform in these anxiety-like tests. So because the HPA axis is overactive in depression models and research, we have expected to see a greater difference in depression-like behavior in these mice. And that's kind of what we do notice. So in the sucrose preference test, our KCC2-CRH canic acid injected mice drink less sugar water, indicating that they're more anhedonic, and therefore this is indicative of a more depression-like state. The nestlet shredding test is another test that looks more at goal-directed behavior. So when you introduce a nestlet into a cage, the mice will, within an hour, will shred their nestlet up into like this nice little nest where they can sit and be safe and protected. If the mouse doesn't shred their nestlet, it's kind of a sign of loss of goal-directed behavior or apathy. And so we see this very robustly in our KCC2-CRH mice. Over a time of 7 days, they generally just don't shred their nestlets, and this is indicative of them having a loss of goal-directed behavior. So we think this is a sign of depression-like behavior. So overall, our data suggests so far that the HPA axis overactivation contributes more to a depression-like phenotype than it does in anxiety. And so the next thing we wanted to test was, how does hyperactivation of the HPA axis cause a more, does it relate to a more severe seizure phenotype? So when these mice were first generated in our lab, Jamie characterized their seizure susceptibility and found that overall, these mice exhibit increased epileptiform activity. So chronically epileptic KCC2-CRH mice injected with pilocarpine, they have an increase in the number of interictal spikes per day. They have an increase in the number of spike clusters per day, and they also have a greater number of ictal events per day. So I wanted to test how these mice performed under the intrahippocampal canic acid model. So I've started to characterize the seizure susceptibility profile for these mice. So these mice, once again, have undergone the ventral intrahippocampal canic acid treatment, and at the same time, they also get implanted with an EEG head mount, and they get hooked up to EEG 7 to 10 days after status epilepticus. And I want to give a mid-presentation shout out to my lab mate, Pandalise Antonodiou. He developed an awesome seizure detection program, so I don't have to torture my eyes with having to score through thousands of hours, multi-terabytes worth of EEG data. So thank you, Pandalise. So here I've graphed both male and female mice. So the male mice are in blue and the yellow mice are in yellow, or the female mice are in yellow. Once again, the solid colored bars indicate the Cre-negative wild-type mice, while the pattern bars indicate the Cre-positive mice. So as you can see in the male mice just alone, The Cre positive or the KCC2-CRH mice exhibit a greater number of seizures per day, so they have a higher seizure burden compared to our Cre negative wild type mice. But interestingly, in our female mice, our KCC2-CRH mice, so the mice that lack KCC2 and CRH neurons, tend to exhibit a decrease in the number of seizures per day compared to the Cre negative wild type mice. This isn't significant, but the trend is pretty apparent, and this is suggesting that potentially HPA axis dysregulation may manifest differently between the sexes. If we look at the average seizure duration, in general, within each sex, the males don't have a difference in the average seizure duration and the females don't either. But it is interesting to note that between sexes, the female mice tend to have shorter seizure durations, once again indicating that these mice just might have a different regulation of the HPA axis. The other thing that we've noticed is that our KCC2-CRH mice have a very robust SUDEP phenotype. So here plotted the survival probability between our wild type Cre negative mice and our Cre positive KCC2-CRH mice, and you'll see that there's almost a significantly increase in the number of SUDEP incidents in the KCC2-CRH mice. And so this is just a representative trace that I've captured for mice that have died of a SUDEP incident. So they will just have this massive seizure event and then they will succumb to their seizure. And the potential contribution of the HPA axis dysregulation in SUDEP is very promising, especially since causes and the mechanisms underlying SUDEP are largely unknown. And so SUDEP occurs, the incidence for SUDEP is about 1 case for every 1,000 in the epilepsy population. 21 to 40-year-old male people with epilepsy are the ones with the highest risk of SUDEP. And while individual risk factors vary, SUDEP incidence occurs highest in those with refractory epilepsy. And the current thought is that SUDEP might be due potentially to a compromised cardiorespiratory function. Finally, most of the current literature right now for SUDEP is focused mostly on genetic models. So the fact that the HPA axis dysfunction can contribute to SUDEP is a novel finding and could inform therapeutic options for people with epilepsy. So we wanted to see if we could attenuate HPA axis function and reduce the SUDEP incidence in our mice. So to do that, we used RU46, which is a glucocorticoid receptor antagonist. The Dancer lab has previously shown that RU46 treatment can reduce status epilepticus-induced hippocampal pathology. So here, compared to vehicle-treated status epilepticus mice, RU46-treated mice show a reduction in pathological ectopic dentate hilus cell proliferation, and that's quantified here. And also, after status epilepticus, there's an increase in the loss of hilar cells, existing hilar cells in the dentate hilus. And when the Dancer lab introduced RU486, there is a reduction in this loss. And so we wanted to try this drug in our KCC2 mice to see if we could attenuate HPA axis function. Could we attenuate the seizure severity that we see in our KCC2 mice? So mice were implanted with a 21-day slow-release RU46 pellet at the time of canic acid injection and EEG head mount implantation. I hooked them up to EEG between 5 to 7 days after status epilepticus, and then we did 2 weeks of continuous EEG. I measured the circulating port levels 12 days after status epilepticus, and you can see that with RU46 treatment, there's a general reduction in circulating port levels, so it has an impact on the HPA axis. And RU46 treatment in our KCC2-CRH mice reduces the number of seizures that the mice have per day compared to our sham-treated canic acid mice. So this is indicating that if we can attenuate the hyperactivation of the HPA axis, we can reduce seizure severity in these mice. And most excitingly is that so far, treatment with RU46 in our KCC2 mice seems to abolish the SUDEP phenotype. The ends are a little low at the moment, but this trend is very exciting. So overall, the data suggests that pharmacologically attenuating the hyperactive HPA axis in these KCC2 mice, we can reduce the severity of their epilepsy. So the conclusions are seizures activate the HPA axis through the downregulation of KCC2 in CRH neurons in the PBN. And so we generated mice that lack KCC2 in CRH neurons, and these mice exhibit an exaggerated seizure-induced activation of the HPA axis. They have increased vulnerability to anxiety and depression-like behavior, and they have this robust incidence of SUDEP. And at the moment, when we pharmacologically attenuate HPA axis dysfunction with RU46, we're able to reduce seizure frequency and abolish the SUDEP incidence. And so the data so far suggests that modulating the HPA axis in epilepsy could help decrease comorbid depression. It could help with mitigating seizure severity and potentially reduce risk of SUDEP. So I've got quite a few ongoing experiments going on. First of which is looking at how chemogenetic inhibition of CRH neurons in the PBN alters seizure severity. So the RU46 pellets attenuate HPA axis dysfunction globally. And so we want to see whether we can inhibit the firing of the CRH neurons specifically in the PBN and see if that can alter seizure susceptibility. I've shown some data looking at the female seizure phenotype in these mice, but I'm currently building up the ends to look at how HPA axis hyperactivation can impact the behavior and the seizure severity, and also looking at their SUDEP predisposition. We're also currently looking at how canonical neuropathology, such as mossy fiber sprouting and dentate granule cell dispersion, might be impacted with the hyperactivation of the HPA axis in these mice. And I'm also looking at how, whether the degree of severity of HPA axis dysregulation can contribute to a poorer behavioral phenotype or poorer seizure burden. And finally, one of our collaborators, Carrie Boychuk from UT Health San Antonio, is helping us look at our mice to see if they have compromised cardiorespiratory function that predisposes them to SUDEP. And as of a couple days ago, she reported that we have a cohort of mice, so we're looking forward to getting some potentially exciting data from that. So with that, I want to thank the lab, Jamie McGuire. I want to thank Pandalise Antonadieu for helping me with the seizure detection program. I also want to thank our collaborators, Carrie Boychuk and Steve Moss. With that, I'll take any questions.
That was great, Trina. That was interesting. Do we have any questions? I have a couple, but I want to see if anybody wants to go first. You can unmute, or you can put it in the chat if you want to, and I can read it to her if you're shy.
I have a question.
Awesome.
Hey, Trina.
Hey, Trina.
So I might have missed this, but what Cree line are you using to obtain the KCC2 CRH?
So we crossed our Phlox KCC2 colony with the CRH Cree colony.
Okay. And do you know when the Cree kicks into your line developmentally?
Developmentally. So I can't answer that. I don't know.
Okay. The only reason why I'm saying this is because, as you know, KCC2 has very important developmental roles and has been shown to alter sort of spontaneous calcium activity during development, affect dendritic formation, synaptic formation. So I wonder how much of the effects that you see are due to changes in connectivity of your CRH neurons compared to the KCC2 effect in the adult.
Yes, so that's actually something that we would like to explore, like network connectivity dysfunction in these mice. And at the moment, I can't answer your question about whether it's developmentally regulated and how that may predispose the adults, but that's something that we want to explore.
Awesome.
Hello, Trina.
Hey, is that Avatar?
It's me. Hey, that was a fantastic talk. I thoroughly enjoyed it. Wonderful. So first things first, say hi to the gang. Second thing is the question. Can you comment on your thoughts on the mechanisms of KCC2 downregulation?
I thought you would ask that. We are starting to explore that. There is evidence in the field that suggests that KCC2 downregulation could be due to to increased BDNF levels. Drawing back to my grad school days, my PhD research, I know that KCC2 is regulated by RE1 cytosine transcription factor, or REST. And so I currently have primers in the freezer waiting to be used and tissue to look at to see how REST regulation of KCC2 might be impacted in these mice. So avenues to explore, no answers yet.
It's very exciting.
Trina, could I ask a question? It's Catherine from Illinois. So great talk. Obviously, I'm very interested in your work, as we know. So I was wondering, I think the earlier data you showed from the 2013 paper, if I remember correctly, it was in the IP canic acid model. And I was just wondering if either you or anyone else in your lab has sort of followed up to see in the ventral IHKA mice, if the CRH neurons show hyperactivity or altered response to GABA receptor activation.
I think that's ongoing. So I have not done that. And I'm not sure if Jamie has preliminary data showing that. I don't think she does. So short answer, at least for now, is no.
Trina, I'll ask a question if that's good.
Yeah, of course.
Nice to see you again and great talk. Congrats on a really nice body of work. When you did the pie charts, you kind of started to separate some of the resilient versus susceptible animals. And I'm curious whether there was some sex differences, because later part of your talks focused on that. And I'm curious, like behaviorally in terms of depressive and or anxiety phenotypes, whether you started to notice there's like specific animals that were more resilient, whether it be males or females.
Right. So this is something that we're actually really interested in looking into because major depression disorder occurs at rates 2 times higher than that of males. And we want to see if that is also something that's reflected in the epilepsy population. Unfortunately, my N's are way too low in the female mice to be able to generate a more conclusive solution, I guess, or conclusion. So I can't really comment on that, but that is something that we're looking into right now. So
cool. Good to see you. And thanks so much.
Yeah, see you, Justin.
So I'll ask a question. So I was thinking about the seizure data in the SUDEP, and I'm trying to think about it. Like, is it that the animals are having like longer seizures, like more severe, and that's just driving, say, some cardiac problems? Or do you think it's a cortisol, like you get jacked up cortisol and so they're responding to a seizure poorly? What do you think might be going on there?
So that's something we're trying to understand. So if you remember, the mice, compared to at least the Cre-negative wild type mice, within each sex, there isn't any difference in their seizure durations. And so it may not be, so we also have to look at the number of seizures. So they are having significantly more seizures, but they're not longer.
They're not longer. Yeah.
And how this contributes to the potential cardiorespiratory dysfunction is something that we're still working on. So hopefully we'll get some more answers with Carrie Boychuk's data. But systemically, because they do have a higher activity of the HPA axis, this might just have a more systemic role in their SUDEP incidence. So something that we're still currently exploring, but we don't quite have an answer to that yet.
Yeah, no. And so with the Cree mouse, is it specific to the HPA axis, or are there, because there's CRH neurons kind of all sprinkled throughout? Are you hitting all of those with the KCC2? Is there not KCC2 in some of the other CRH cells, or? So
the idea is that we're hitting those that all CRH neurons that have KCC2.
All CRH neurons. Okay. So
that's why we're gonna start looking. I have started looking at injecting the dreads, the GI dreads, specifically in the PBN to more localize the mission.
Do we have any more questions? Going once, going twice. Thanks, Trina. We had a bunch of questions. I think this is really interesting. It's a really good area of research, and I think you're doing a great job with it. So thank you very much for joining us, and I hope everybody had a good time. And until next time, everybody.
Thanks for having me.